Now let's focus on digestion of the fat component of the meal as it travels down the alimentary canal. As the food particles are mechanically broken down in the mouth, cells in the tongue secrete the enzyme lipase. This enzyme begins to process the fats, also called lipids, chemically. However, major chemical digestion of fats does not begin until the food reaches the small intestine. After peristalsis propels food particles into the stomach, muscular churning helps break them up. Because the fats are insoluble in water, they tend to aggregate into large globules as they're churned. The pancreas secretes several enzymes into the small intestine, including the enzyme pancreatic lipase. Lipase breaks down fats. However, before the molecules in the fat globules can be digested by enzymes, the globules must be broken up or emulsified. Bile, secreted by the liver and stored in the gallbladder, contains small lipids called bile salts that emulsify fat globules. The gallbladder releases the bile into the small intestine. Fat globules from food, lipase from the pancreas, and bile salts from the liver come together in the small intestine. When bile enters the small intestine, the bile salts emulsify the large fat globules into small fat droplets. Pancreatic lipase breaks up the small fat droplets into monoglycerides and free fatty acids. Once fatty acids have been liberated, it's thought that fatty acid binding proteins bring them through the microvilli of the cells lining the small intestine. There, they're processed into protein-coated complexes and released into the bloodstream for transport to fat storage cells and other tissues throughout the body. Again, the undigested waste is eliminated via the large intestine.